Hello everyone, I'd like to thank you for joining us today for this webinar. My name is Erica Chow and I'm with BCS Incorporated. I currently provide support to the Bioenergy Technologies Office, or BETO, and will be moderating the webinar today. To begin the webinar, I will provide a brief overview of the Bioenergy Technologies Office, which will be followed by a presentation from Dr. Richard Sayer, representing Los Alamos National Laboratory and the New Mexico Consortium. Please record any questions and comments you may have during the webinar and send them to eere_biomass@ee.doe.gov. As a follow-up to the webinar, the presenter will provide responses to selected questions. All slides from this presentation will be posted online at the following web address. For general questions regarding the Bioenergy Technologies Office, please email the same address listed here. As some additional background on today's webinar, I'd like to remind everyone that this is part of the BETO ongoing webinar series. Our webinars cover many of the office's activities and feature hot topic discussions that are relevant to the development of renewable fuels, power, and products from biomass resources. More information can be found on our website listed here. To provide more information about the Bioenergy Technologies Office, we've outlined some key points here on our vision, mission, and strategic goals, which include helping to transform the nation's renewable and abundant biomass resources into cost-competitive, high-performance biofuels, bioproducts, and biopower. BETO supports ongoing research and development efforts to help create robust next-generation domestic bioenergy industry in the United States. And by doing this, the Department of Energy supports the effort to provide Americans with sustainable, renewable, alternative energy resources. These efforts enhance U.S. energy security, reduce our dependence on oil, provide environmental benefits, including reduced greenhouse gas emissions, and create economic opportunities across the nation. BETO's mission is to support research, development, demonstration, and deployment of advanced bioenergy. This slide here lists BETO's core focus areas, and they include feedstock supply, efforts to focus on R&D to develop cost-effective integrated logistics systems. So this includes growing, harvesting, collecting, storing, pre-processing, handling, and transporting quality feedstocks to biorefineries. Our conversion R&D programs is focused on developing commercially viable technologies to convert terrestrial algal feedstocks into biofuels, as well as bioproducts and biopower. The demonstration deployment program is focused on demonstrating and validating biomass conversion technologies through successful construction and operation of cost-shared pilot, demonstration, and commercial-scale integrated biorefineries. We have cross-cutting programs in sustainability analysis, proactively addressing sustainability of advanced bioenergy to enhance its benefits, public acceptance, and long-term viability. So now I'd like to turn the floor over to Dr. Richard Sayer of the Los Alamos National Laboratory and the New Mexico Consortium to provide his portion of today's presentation on genetically modified algae, a risk-benefit assessment.
Hello, I'm Richard Sayer. I'm a senior research scientist at Los Alamos National Laboratory, and it's my pleasure today to present some of the work that we've been focusing on addressing the risks and benefits of genetically modified algae for producing biofuels as well as high-value co-products. On the next slide is an outline of the presentation that I'll be giving today. We'll start with a historical perspective, and that focuses on how have GM traits been managed in the agricultural sector? What are some of the known potential benefits and risks of GM traits in crops? And how are those agricultural traits managed? Then we'll shift to the special case of algae, and we'll start with what is the potential for algae in meeting renewable fuel requirements? What is the role of genetically modified traits in producing algal biomass? What are the perceived risks for genetically modified algae? How can those risks be mitigated? what agencies are currently regulating the release and commercialization of genetically modified algae. And then finally, I'll present some examples of emerging algal genetically modified traits from our group that focus primarily on crop improvement, and then summarize the presentation. So on the next slide, we are looking at some of the historical issues surrounding genetically modified plants for agriculture. And the primary driver that has really pushed this industry is increased income generation. And as you can see, the increase in crop value from genetically modified traits has increased by $98 billion since their initial release in 1996 through 2011. GM technology, importantly, is also a part of a sustainable solution for agriculture. And as a result of the adoption of GM crops, there have been substantial reductions in carbon dioxide emissions as well as in pesticide use. A large part of this reduction in CO2 emissions and also pesticide use is associated with the ability to do no-till agriculture. And no-till agriculture, particularly in soybeans, was really made feasible by the introduction of herbicide-resistant traits. Very importantly, in the United States, it's been estimated that over 1 trillion meals of GM foods have been served since 1996. And to date, there are no known illnesses that have been demonstrated from the consumption of genetically modified foods. So there's an incredibly strong safety record for the management and utilization of genetically modified foods. In addition, there's been increasing international adoption of genetically modified crops, and most importantly in developing countries. And in fact, in the last 2 years, the acreage in GM crops in developing countries has actually exceeded that in the developed world. And it continues to grow at a relatively high rate of about 6% increase in acreage per year. However, we face some emerging challenges, particularly with respect to global food security. And it's in this context where GM technology perhaps will have its greatest benefit. I'm going to give 2 examples that support this position. The first being the C4 Rice project that's funded by the Bill and Melinda Gates Foundation. This project is managed at the International Rice Research Institute in the Philippines. And as a result of the work that they do at Erie, analyzing the natural genetic diversity within their vast collection of rice varieties as well as wild relatives, they've come to the conclusion that by 2050, the genetic variation that can be utilized for crop improvement will be exhausted. And so to continue to provide food roughly for about 1/3 of the world's population that consumes rice, they really need a quantum leap in crop production. And that's led to the funding of the C4 Rice project. The objective of that project is to engineer a C3 low-efficiency rice crop into a high-efficiency C4 rice crop. Closer to home, the BioCassava Plus project, also funded by the Bill and Melinda Gates Foundation, and for which I was the principal investigator, actually came to the conclusion that to biofortify cassava to meet the minimal iron requirements in a cassava-based diet could only be achieved by genetic modification, that the genetic variation for iron content in cassava roots was insufficient to actually hit our target value. And this conclusion was actually reached by another project funded by the Gates Foundation called Harvest Plus. So there are opportunities for GM technology to address major challenges facing the future for food security. So if we look at some of the drivers and benefits for transgenic crops, particularly in the U.S. As I mentioned earlier, income generation is probably the most significant driver, and that's due to the lower cost of growing GM crops compared to non-GM crops. In addition, we have more sustainability growing many of the GM crops relative to non-GM, associated with reduced energy usage in no-till agriculture and the reduced use of broad-spectrum synthetic pesticides. Associated with the use of no-till agriculture, of course, is reductions in soil erosion. And so again, in many cases, GM crops can actually provide increased sustainability relative to non-GM crops. But in the future, particularly in the developing countries, we will see applications of GM technology to provide better nutritional composition for foods, extend the shelf life of foods so that they can be marketed into urban areas, increase stress tolerance, and particularly drought and heat tolerance, which are emerging considerations associated with global climate change, the production of renewable green chemical feedstocks, including biofuels, and perhaps pharmaceutical productions in pathogen-free organisms that don't support the growth of pathogens that can infect humans. So, of course, there are risks associated with any technology, and it's the balance between the benefits and the risks that help us decide whether the technology should be put forward or not. With respect to transgenics, of course, one of the challenges we face is that currently transgene introduction into plants and also in algae in most cases is a random event. And so there may be unintended mutations associated with the introduction of the transgene that are potentially undesirable. Fortunately, in most cases, those are readily identifiable, particularly through genome sequencing approaches, but also phenotypic analyses. Another concern, and a serious concern, is transgene escape to related plants and pollination of nearby relatives. In many cases, this issue is managed by limiting the release of transgenic crops only to those areas where progenitor species are not native. But in some cases, that's not possible. And there are examples of herbicide resistance genes moving from canola, an oilseed crop, into other related weedy species and resulting in herbicide resistance in those weedy species. Another concern is increased use of herbicides. So one of the predominant traits in GM agriculture is herbicide resistance to control weeds. And associated with that, of course, is the increased use of herbicides. So ideally, herbicides that should be used for weed control should be of low toxicity. And then, as I mentioned earlier, is the development of herbicide-resistant weedy plants. Another concern that's been expressed, particularly by the non-scientific community, but also within the scientific community, is the concern about genetically uniform strains being planted as a result of the introduction of GM traits. Now, those two events are not linked necessarily. So the introduction of GM trait does not have to be associated with a genetically uniform crop. But, of course, there are concerns about using genetically uniform crops, particularly with respect to disease and stress tolerance. There are also potential unanticipated alterations in food composition. And this really gets back to the first point, and that is that there is the potential to do mutations, perhaps in pathways that are critical for the production of molecules that are important in nutrition for food consumption. But another concern is that there may be alterations in metabolic flux associated with genes that are unintended. To address that concern, typically what is done is to compare the metabolite composition of GM foods to non-GM foods to see if there are alterations in the composition. Another concern is the expression of new allergens. And so there have been examples of strategies for introducing proteins that have enriched composition of essential amino acids that potentially have come for allergenic proteins. Fortunately, in the review process, these issues are recognized, and they're now standard approaches using bioinformatic tools to identify potential allergenic domains and primary protein sequences. There are also downstream problems, and that is primarily the segregation of transgenic from non-transgenic crops. And so particularly in the European community, there are regulations about the potential contamination of non-transgenic crops with transgenic crops. And so that introduces an important step in the process of growing and harvesting and distributing these crops, and that is a demonstration that they're not non-transgenic crops are not contaminated with transgenic crops. And then because of the high costs of commercializing transgenic crops, there tends to be greater market control by fewer and fewer producers. So to manage these risks and benefits, in the United States, there are a number of regulatory agencies that can be involved at different stages in the approval process for the testing and commercialization of GM crops. The examples that I'm going to give here are some of the issues that we had to address in the BioCassava Plus program to not only conduct field trials in the United States, but also in Africa under the most stringent requirements of the Cartagena protocols. And so primarily the first issue that we need to address with transgenics in the United States is the issue the issues addressed in the Plant Protection Act, and that is no sequences can be introduced from pathogens or of human origin into plant materials. Generally, that's not a challenge. The next issue is, are transgene products potentially toxic or allergenic? And so would the enzyme, for example, that may be the product of a transgene produce a toxic molecule, or could itself be allergenic? And as I mentioned earlier, there are bioinformatic strategies that can help address those concerns. Another aspect is preventing the introduction of non-essential DNA sequences into transgenic plants. So only those sequences involved in the expression of the transgene ideally should be introduced into the transgenic plant. Again, as I mentioned earlier, the integration of the transgene in the plant genome should have no off-target effects and yield a nutritional composition, unless it's enhanced, in the transgenic plant should really be identical or have no substantive alterations relative to wild-type plants. The next stage is the conduction of confined field trials, and these are generally conducted under nationally or internationally recognized protocols. Some of the examples of those safety concerns that we had to address through the Cartagena protocols include fencing off the field trial areas, providing continuous 24-hour surveillance, and leaving the fields to lie fallow for at least a year after the trial is finished to ensure that no volunteer organisms or transgenics reemerge after the field trial is finished. To reduce the likelihood of transgene escape, all flowers have to be systematically removed so that the possibility of pollen or seed dispersal is greatly diminished. And animals that potentially could disseminate seeds or portions of the transgenic crop also have to be restricted from the field crop trial area. Finally, at the end of the field trial crop, all of the plant material must be destroyed, and typically that's done by burning all of the residue in a deep pit, followed by covering that material with soil. So after the field trials are carried out, ideally, of course, the results should show no consequential or unintended impacts on the yield. As I mentioned earlier, the food composition and or the environment. The next stage is then taking the food material, if this is a food, and conducting animal feeding trials to demonstrate that there are no adverse effects on animal nutrition or health. If those feeds or food materials are ultimately going to be consumed by humans, then the next stage is to demonstrate the complete digestion of the transgenic protein in an artificial human stomach, and that no segments of the protein remain undigested after this treatment. After positive results from in vitro trials, then it moves on to human feeding trials, again, ideally showing no adverse effects on health or nutrition, and finally regulatory review and approval. So in total, this is a very involved and well-developed process that has served the transgenic crop community very well. So I'm switching gears and now focusing on algae. The reason that we're particularly interested in algae is, of course, their ability to produce tremendous amounts of biomass in a very small area. And it's been estimated, in fact demonstrated in a variety of trials, that algae potentially can produce anywhere from 2 to 10-fold more biomass per unit land area per year than terrestrial crops. And so we're looking at exploiting that very high productivity and rapid growth to produce renewable fuels and also high-value co-products. So as indicated here, algae have high photosynthetic efficiency. Every cell in microalgae is actually photosynthetic, unlike crop plants where there are tissues such as roots and stems that may not be photosynthetic that have a photosynthetic demand. They're capable of very rapid division, and importantly, they can have high lipid contents as shown in the figure. In the figure, the lipid droplets are visualized using a fluorescent dye that is shown here as yellow fluorescence. Importantly, we can also recover all the biomass during harvesting, and we're not leaving behind residues in the field, as is the case with terrestrial crops. And we potentially can harvest algae throughout the week, throughout the day, and perhaps throughout the year. This is important because it substantially reduces risk. So in terrestrial crops, there is the possibility of having crashes that can be associated with adverse weather or disease, for example. And then one may have to wait a year before another crop is grown. In our experience in cultivating algae, that when we have crashes, generally these can be mitigated very rapidly and a new production system put online within a matter of days. And very importantly, algae are perhaps more sustainable means of producing biofuels than terrestrial crops. And in part this is associated with the ability of algae to capture carbon dioxide in ponds as bicarbonate. So CO2 generated at point sources can be injected into ponds, hydrated to form bicarbonate, which is non-gaseous, and bicarbonate is actively pumped into the algae to use as a substrate for photosynthesis. In addition, we can use wastewater to provide nutrients, and there's no direct competition with food if we choose our sites to grow the algae in non-agricultural areas. So why is GM technology being considered as part of the solution for algal crop improvement? In part, this is because at the present time, we don't have breeding systems in place for commercial algal strains. Chlamydomonas, which is more or less the lab rat of algal molecular biology is the dominant generation is haploid, and it is possible to do mating and look at gene segregation and actually to breed for improved phenotypes. But those breeding systems have not been developed for most commercial strains at the present time. And so we're left with other strategies to try to improve the biology and the productivity of algae. Those include bioprospecting efforts to identify better strains in the wild than the current commercial strains, using mutagenesis approaches to generate new phenotypes that are high value, and the introduction of GM traits. All three of these are perhaps our best strategies currently available for improving the biology and the performance of algae. In addition, GM traits or GM strategies can be used to introduce new biochemistries and physiologies into algae, for example, to produce high-value co-products in high volumes. And this can help substantially reduce the cost of producing fuels. And then finally, GM algae, it's possible to grow them in contained fermentation systems to reduce the chance of escape. And so biocontainment of GM algae is therefore feasible, unlike terrestrial crop plants. So there are a number of potential risks, of course, associated with the introduction of GM traits into algae. And some of these risks are actually unique to algae and are different than those risks that we need to consider with respect to terrestrial crop plants. One of those risks is aerosolization and the global spread of algae. Being very small microorganisms on the order of a few microns, they're very easily suspended into the atmosphere during aerosolization. And in fact, it's known that algae sometimes travel great distances across the globe through the jet streams. Another concern is their persistence. Many algae can survive very long periods of time completely desiccated in soils and then re-emerge once water is available. Another concern is weedy or invasive traits. If we introduce traits into algae that enhance their growth and competition with other algae in the wild, if they have increased ability to take up nutrients and outcompete other wild algal organisms, if they produce toxins or express molecules that provide evolutionary or competitive fitness, then we have the potential for algal blooms of GM-modified algae that may alter species compositions in the environment. And of course, there is always the concern of gene escape to related species in the community. Another issue in common with agricultural GM risks is the issue of health impacts. And so it's generally accepted within the algal community that antigenic proteins will be managed, as well as limiting the production of toxins. And finally, there is the concern for mitigating the potential for horizontal or sexual transfer of transgenes. This is particularly a concern in cyanobacteria, which are known to be promiscuous in terms of their gene exchange capabilities. So what are some of the potential environmental outcomes? So in this particular figure in the upper left box is a distribution of algal species as indicated by the different colored lines. So in a natural community, there's often a diversity of algal organisms growing in concert. In the upper right corner is an example of what we would ideally like to have in a pond, and that is with a genetically modified organism, a monoculture essentially of highly productive and high-yielding GM algae, either producing molecules for biofuels or high-value co-products. If, of course, we have escape from that pond, then in the lower left box, we see a situation where there's no consequence of the GM algae escaping, and that is the species composition is not altered by the release and the growth of a genetically modified organism. And that's indicated by the yellow line in the lower left corner of the box. And you can see that the population dynamics, or the biomass of the GM algae, is transient and does not persist or alter the species composition in the wild. The worst case scenario is the lower right box. And in that case, the GM algae has increased environmental fitness relative to the natural populations and dominates the species diversity in that environment, and perhaps could lead to a harmful algal bloom if sufficient nutrients were available to allow it to grow very rapidly. So that's the situation that, of course, we want to avoid and to prevent from happening by the appropriate regulatory constraints. So ideally, GM traits should have a minimal impact on the environment. They should reduce the evolutionary fitness of the algae in the wild so that they're not strong competitors with wild algal species. As I mentioned earlier, the algae should not produce toxins or have traits that are potentially harmful to living organisms or disrupt ecosystem health. It's critical that we evaluate the risk potential, since in many cases we don't or cannot predict the outcomes of GM traits in the wild. And to do so requires controlled field trials with multi-species components, and these need to be carried out to evaluate the potential for risk. And then finally, another strategy is to introduce biocontainment traits into genetically modified algae to reduce their potential for gene transfer. Some examples of those strategies include stacking conditional lethality traits that are perhaps complemented by exogenous molecules in a pond, the inactivation of genes that control sexual transmission to reduce the potential for gene transfer, and the expression of terminator genes that are activated upon escape from a controlled environmental growth situation. So with respect to the latter trait, what we're showing here are some of the recent results from work that we've been doing in our group to introduce biocontainment traits into genetically modified algae. In this case, the gene that we're introducing is a gene called barnase. And barnase is a general ribonuclease that will degrade the RNA within the cell. Now, fortunately, barnase can be inhibited by a protein called barstar, which has a very high affinity for barnase. And barstar, in turn, in our case, is going to be regulated under the control of a gene switch. So this gene switch, in turn, is regulated by caffeine. So in the context of this application, we're growing algae in the presence of caffeine, and under those conditions, barstar gene is expressed and inhibits the barnase ribonuclease activity. When caffeine is not present, for example, if the algae were to escape a pond that contained caffeine and go to an environment where there is no caffeine, then the barstar gene is inactivated, and the barnase activity in turn takes over and limits the growth of the cells. So we've had good success with this technology. We're getting substantial suppressions and growth in the absence of caffeine, and we're now modifying that to eventually show no growth in the absence of caffeine. So it's this kind of approach, potentially using terminator gene technology, that's regulated by a xenobiotic that could only be found in a pond environment, that may allow us to contain genetically modified organisms. So moving on, I want to briefly discuss some of the regulatory environment addressing the release and commercialization of genetically modified algae. I'd also like to direct your attention to the bottom of the slide for a slideshow that was presented by Dr. David Glass at the recent International Algal Biofuels, Biomass, and Bioproducts Conference in Santa Fe in 2013. And in David's presentation, he goes into a thorough coverage of the regulatory environment for genetically modified algae. So very briefly, GM algae is regulated by the EPA under the Toxic Substances Control Act, or TSCA. And if an organization anticipates doing a field trial or an outdoor release, the process involves making an application to the Environmental Protection Agency under the TSCA experimental release application process that undergoes a review and hopefully an approval, and that takes about 60 days advance notice for that process to work through. For commercial activity, there's another regulatory approval process called the Microbial Commercial Activity Notice, or MCAN, and that requires an additional period of notification for EPA review and approval. In addition, other regulatory agencies besides the EPA, including USDA and FDA, may have regulatory approval required for the release or commercialization of GM algae. And that will depend on the types of products that are produced and their particular use. And again, I refer you to the presentation by David Glass. So finally, I want to mention some of the work that we're doing on algal biofuels. And this is more or less, on this slide, is a summary of some of the life cycle and economic analysis that was carried out by the National Alliance for Advanced Biofuels and Bioproducts. That program began in 2010 and finished in 2013. So in 2010, we have a base case scenario where algae, generic algae, were being grown in open ponds. They were harvested by centrifugation. They were typically dried and wet solvent extracted. There was no nutrient recycling. And then the oil was converted into typically diesel by transesterification. So we modeled that base case scenario as part of the NAB program for a large-scale production facility in Pecos, Texas. And the estimated cost of producing fuel from that base case scenario ranged from $230 to $16, so non-competitive with petroleum at that time. After 3 years in the integration of the best technologies that we could identify through the NAB program, we're now approaching $8 a gallon, still higher than $3 a gallon for gasoline gallon equivalents from petroleum, but a substantial improvement over the base case scenario in 2010. The two factors that are going to have the highest impact, though, on reducing the costs in the future were also identified, and that is productivity of the algae and the costs associated with harvesting those algae. To move the price from $8 to $3 will require at least a threefold increase in productivity over the generic or base case scenario in 2010, and also a substantial reduction in the harvesting costs of producing those algae. And this is a case where genetic modification can have a substantial impact by increasing the yield or the productivity of the algae. So in our group, we've been trying to improve the biomass productivity of algae by increasing the apparent efficiency of photosynthesis. And on this figure that's drawn from a review article by Zhu et al., one can one can divide the various inefficiencies in photosynthesis into three major problem areas. The first being light capture, the second is the energy conversion or the biochemistry, and the third factor is energy accumulation. And it's apparent from this diagram that most of the losses in efficiency actually occur in the light capture and its conversion actually into charge-separated states. The overall efficiency of photosynthesis, however, in crop plants is very low, ranging from 4 to 6% on this particular figure, but oftentimes in the field it's even less than that, in the range of 1 to 3%. So there's substantial room for improvement. Theoretically, based on thermodynamic evaluations, it's potentially feasible to increase the efficiency up to 11% using solar energy. If we use the energy of red photons, even higher, up to 30%. So there's substantial room for improvement in photosynthetic efficiency. So our strategy was really to address the constraints around light capture and energy conversion in the photosynthetic electron transfer complex. And as indicated by the red here, the maximum rates of photon capture at full sunlight are actually 10 times faster than the maximum rates of electron transfer. And that's due to the fact that there's a kinetic bottleneck between photosystem 2 on the left and photosystem 1 on the right, which is the cytochrome B6F complex. And so this is the slowest step in the electron transfer process and has a rate constant ranging anywhere from 1 to 10 milliseconds. So the rate of photon capture is 10 times faster than the rate of electron transfer through the cytochrome B6F complex. So our strategy to address this challenge of the kinetic bottleneck in electron transfer was not to improve the efficiency of electron transfer through the cytochrome B6F complex, but to actually reduce the efficiency of light capture, to better couple the kinetics of light capture by the light harvesting antenna complex to the downstream kinetic bottlenecks in electron transfer. So on the left side of the diagram is shown the typical architecture of light harvesting antenna and photosynthetic complexes of plants in the canopy architecture or in algae as they grow in a pond. And that is all of these leaves in the case of a tree, for example, in a canopy architecture, or the individual algal cells have very large antennae. And so the leaves or the algae at the top of the canopy or the pond are capturing at full sunlight intensities many more photons that they can actually convert into charge-separated states. And so up to 90% of the energy that's captured by the antenna is actually dissipated as heat or fluorescence. And of course, the light intensity is diminished as one goes down in the canopy, and at some point the antenna actually are too small to actually capture photons to carry out photosynthesis. Now this particular antenna architecture actually works very well in mixed species environments where light is actually a resource that needs to be captured more efficiently by any given species than your competing species. Otherwise, you're likely to lose in that competition. So having a large but inefficient antenna actually provides a selective advantage because the organisms can restrict access to other competing organisms to light. Now, in a pond environment, ideally we'd like to capture every photon and use every photon to do photochemistry and to drive photosynthesis. So on the right side is a different type of architecture for antennas. And in this architecture, we have smaller antennas. And if it's a fixed canopy architecture, such as in a forest, smaller antennas at the top of the canopy and larger antennas at the bottom. And so less energy is lost, more energy penetrates deeper into the canopy or the pond and can ultimately be captured by the antennas of algae or leaves lower in the canopy. To test this hypothesis that smaller antennae may actually increase photosynthetic efficiency, we decrease the size of the antenna in transgenic algae by inhibiting the production of chlorophyll B. Chlorophyll B is one of two species of chlorophyll found in higher plants and in green or chlorophytic algae. And chlorophyll B's function is only to serve in the antenna. Chlorophyll A is found in the antenna, but also in the reaction centers where charge separation occurs. And so by limiting the production of chlorophyll B, specifically, we can actually reduce the size of the antenna without directly impacting the reaction center where charge separation occurs. And what we demonstrate in this particular diagram is the rate of photosynthesis as a function of the size of the antenna. And here we're measuring the size of the antenna by the chlorophyll A-B ratio. So as we reduce the amounts of chlorophyll B, the A-B ratio increases. In this case, we see two algae with intermediate size antenna having chlorophyll A-B ratios of roughly 5 and 4, an algae that has no chlorophyll B, this is called the CBS3 mutant, so it has a very small antenna, and then wild-type algae with very large antenna. And it's apparent here that photosynthesis in wild-type algae is saturating around 450 micromoles of photons per meter squared per second. That's about 1/4 of full sunlight intensity. In contrast, algae with no peripheral antenna complex, such as CBS3, have higher rates of photosynthesis at higher light intensities, whereas the intermediates actually do better than the very small antenna or the very large antenna. Now, these rates are expressed on a chlorophyll basis, and so they're somewhat marginalized by a small reduction in chlorophyll, but also on a cell basis we see the same trends, and that is algae with intermediate size antennas have higher rates of photosynthesis than algae with very small antenna or algae with very large antenna. Does that convert into increase in biomass? And the answer here is yes. Under low light conditions, you can see here that the growth, as indicated by cell density, of wild-type algae and algae with intermediate-size antenna is virtually the same, whereas algae with very small antenna are not growing well at all. Under high light or saturating light conditions, again, the algae with large antenna are doing better than wild-type or the algae with the small antenna. So these algae that I just described have fixed antennas. But of course, light intensity changes throughout the day. It changes throughout seasons. It's a constantly changing input that drives photosynthesis. So ideally, we would like to have antennae that could self-adjust and optimize their photosynthetic efficiency regardless of the light intensities during the season or during the day. And so we've developed a strategy in which we can regulate the size of the antenna as a function of the light intensity in which the algae are growing. So these are self-adjusting antennas. And the strategy here is we start with algae that have no capability of making chlorophyll B. We then introduce the gene called chlorophyll A oxygenase that allows them to make chlorophyll B. But at the 5' end of the gene, we add a 19 base pair light-responsive element to that messenger RNA. That light-responsive element is the binding site for a translational inhibitor called NAB1 that was identified by Olaf Kruz's lab in Germany. NAB1 will prevent the translation of the messenger RNA to which the LRE is fused. NAB1, in turn, is regulated by light intensity. There is abundant and very high levels of NAB1 under high light and low levels of NAB1 under low light. So the light regulation is translationally controlled by the NAB1 protein. And the model predicts that under high light, NAB1 blocks translation of the chlorophyll A oxygenase. Chlorophyll B synthesis is inhibited, the AB ratio goes up, and the antenna size shrinks. The opposite case is low light, and under those conditions, NAB1 levels are reduced. That allows the translation of the chlorophyll A oxygenase messenger RNA. Chlorophyll B synthesis goes up, as well as antenna size, and it would be predicted that the cultures would grow well under low light conditions where big antennas provide an advantage. So that's, those were actually the outcomes of this experiment. So in this particular experiment, we're looking at the growth of algae with different antenna sizes, as well as capabilities to modulate the size of their antenna. So the blue lines are wild-type organisms, including a complemented wild type with the CAO, or chlorophyll A oxygenase gene, but lacking the light-responsive element. And you can see over the course of time that the AB ratio, or the indicator of the antenna size, does not change as culture density increases with time. That's in contrast to those with the self-adjusting antenna. And so in the beginning, when we do have a low inoculation or low cell numbers, there's high light intensity throughout the culture, and the antenna are small. As the light intensity is diminished, as cell number increases, then the antenna size increases, and that's indicated by a reduction in the chlorophyll AB ratio. So here we're actually looking at the rate of photosynthesis, again as a function of light intensity for algae that have self-adjusting antenna. Those are the NAB strains. Algae with no chlorophyll B synthesis and very small antenna, that's CBS3. And then wild type, complemented wild type, and complemented wild type with a CAO gene containing the light-responsive element, but a mutated light-responsive element that would no longer bind the NAB protein. So a number of controls here. And what you can see is that those algae that were indicated in the last figure to have the largest or greatest increases in, or changes, I should say, in antenna size, actually have here also the highest rates of photosynthesis as a function of light intensity, 3 times greater than the wild type strain. So does that translate into increased biomass? And the answer is yes. And in fact, we had a 2-fold increase in dry rate productivity as well as cell numbers over the course of a 12-day cultivation under environmental conditions, mimicking what would happen actually in a pond. And so this approaches our objective of a 3-fold increase required to reduce the cost of producing fuel from $8 to $3. So a simple one-gene transgenic trait that improves the efficiency of photosynthesis, increases biomass productivity, can potentially help us to achieve our objective of reducing the price of fuels generated from algal biomass to levels that are competitive with petroleum. Now, very importantly, we've also demonstrated in separate experiments that these algae with engineered antenna sizes will not outcompete wild-type algae in mixed cultures. And in fact, they lose, and wild-type algae win. And the reason for that, as I've mentioned earlier, is having a large antenna, even if wasteful, allows you to capture more light and exclude it from your competing organisms. And so this is an example of a trait improvement that actually reduces evolutionary fitness in the wild, but as a monoculture, improves the overall yield of algal production. So in summary, what we've discussed today is the potential for algal biofuels becoming economically feasible. As discussed, we need to increase the yield by a factor of 3 over current production rates. We need to have a higher energy return on investment, ideally greater than 10. And we need to reduce carbon emissions and enhance environmental services. In other words, increase sustainability. There are concerns about using GM technology, and perhaps the greatest concern is actually weediness or invasive species that could potentially lead to ecosystem disruption. But many of the traits that will likely be engineered into algae are traits that will also reduce environmental fitness and competition with other organisms. For example, increasing the oil content is known in many cases to actually reduce the rate of cell division. And so there's not going to probably be an evolutionary advantage to having higher oil content in the wild. But to address concerns, of course, it's always wise to be careful, and one approach is to introduce biocontainment traits into algae. Some of those can be through traditional approaches such as mutagenesis, so conditional lethality mutations. Those can potentially be stacked. Introducing mutations that will prevent the capability of carrying out meiosis and diploid production strains, and transfer of genes through mating, or the introduction of terminator gene technologies that are under the control of xenobiotics that are present in ponds but absent in the wild environment. Regulatory processes are in place at the present time, and the Environmental Protection Agency currently has authority over field trials and commercialization of GM algae, but depending on the trait, the U.S. Department of Agriculture as well as the Food and Drug Administration may also have regulatory approval processes. So in summary, there's great potential for improving productivity, producing high-value co-products from algae, increasing the sustainability of algae using genetic modifications, but we have to be careful, and we need to carry out the appropriate evaluations and field trials to be sure that the products that we generate through advanced genetic technologies not only improve the environment, but are safe for the future as well. Thank you.
So thank you, and thank you, Dr. Sayer, for joining us today. As I mentioned before, these slides will be made available on the BIDO website. Please direct any questions to the email address listed here, eere_biomass@ee.edu.